Today we'll be talking about control of prokaryotic genes or bacterial genes. Bacterial metabolism. Bacteria needs to respond quickly to changes in its environment. If they have enough of a product, they need to stop production. So for stop, why? Because it's a waste of energy to produce more, and they stop production of enzymes for synthesis. If they find a new energy source or food source, they need to utilize it quickly for their metabolism, growth, and reproduction. They do this by they start the production of enzymes for digestion of this new food or energy source. Remember regulating metabolism? So there's feedback inhibition. Products act as an allosteric inhibitor of the first enzyme in the tryptophan pathway, but this is a wasteful production of enzymes. So the presence of tryptophan will prevent the production of the other enzymes 1, 2, 3, 4, and 5. But this is wasteful because why are you producing enzymes that you do not need? So a better way, or a different way to regulate metabolism is through gene regulation. Instead of blocking the enzyme function, you block the transcription of genes for all the enzymes in the tryptophan pathway. This saves energy by not wasting it on unnecessary protein synthesis. So the production of tryptophan inhibits the production of genes and mRNA that will then code for these enzymes that will produce more tryptophan. So this is a good example of a negative feedback loop. In the presence of tryptophan, the transcription of gene 1, 2, 3, 4, and 5 is turned off, and the genes are not expressed to produce those enzymes. So the increase of 1, which is the increase of tryptophan, will decrease the transcription of gene 1, 2, 3, 4, and 5 to produce more enzymes of tryptophan. Gene regulation in bacteria. Cells vary in amount of specific enzymes by regulating gene transcription. So either genes are turned on or turned off. An example of turn genes off is if bacteria has enough tryptophan when it doesn't need to make more of its enzyme used to build tryptophan. So that is a turn off, which is an example of a negative feedback loop. In the presence of tryptophan, something else turns off. For a genes on example, if bacteria encounters new sugar like lactose, then it starts to produce enzymes used to digest lactose. This is a good example. This go idea is a good example of a positive feedback loop. So in the presence of one molecule, in this case lactose, it will increase the production of enzymes that are going to digest lactose. So it plus one will increase the plus of the other. So increase lactose, increase the enzymes to digest the lactose. Bacteria group their genes together. So unlike animal and eukaryotic DNA that can be transcribed differently because of the introns and exons, bacteria have something called operons. Operons are how the genes are grouped together, and they label them very nicely. So the tryptophan operon obviously involves tryptophan, which is an amino acid. The promoter is where RNA polymerase will bind. A single promoter controls transcription of all the genes in the operon. So for the tryptophan operon, which is below, when the RNA polymerase binds to this binds and transcribes, you're going to get all of the tryptophan enzymes and proteins that are necessary. It transcribes as one unit and a single mRNA is made. The operator is the DNA binding site of the repressor protein. So the operator is where the repressor protein will attach and will prevent the RNA polymerase from binding and transcribing the DNA. So how can these genes be turned off? Through the repressor protein, like I just mentioned. It binds to the DNA at the operator site, and it blocks the RNA polymerase from binding, which will then block the transcription. So typically, if there's no repressor, you're going to have the DNA, which will be transcribed to your RNA, which will code for a protein. In the picture below, when the tryptophan is present, it is the co-receptor or repressor, so it'll attach to the protein, the active or the repressor protein, and when it attaches, it will then be an active repressor. It will join to the operator site, which is up there in yellow, and no mRNA can be made. So the operon model, you have the operon, the promoter, and genes they control. They serve as a model for gene regulation. So below you have your RNA polymerase at the top box, which is your promoter site. You have your operator where a repressor can bind to prevent the RNA polymerase from transcribing the genes. And when the RNA polymerase runs down the gene, you will produce an mRNA, which will then produce proteins, and in this case, enzymes 1, 2, 3, and 4. When the repressor binds, it prevents the RNA polymerase from binding and transcribing more mRNA. So a repressible operon, an example is the tryptophan operon. It's a synthesis pathway model. When there's an excess in tryptophan, it binds to the tryptophan repressor protein and triggers repressor to bind to DNA. This blocks transcription. So the RNA polymerase will go down the gene. It will transcribe your mRNA. The enzymes will then produce tryptophan amino acid. The tryptophan amino acid will bind to the repressor. The repressor will bind to the operator. And when it binds to the repressor, there'll be a conformational change in the repressor protein where it then binds to the operator, preventing RNA polymerase from binding and transcribing the genes. So that was the example of the tryptophan operon. So what happens in the presence of tryptophan? There doesn't need to be any more tryptophan made, so you don't need to make tryptophan building enzymes. So it stops the production of tryptophan production. Tryptophan is an allosteric regulator of repressor protein. Inducible operon, an example is lactose. The digestive pathway model, when lactose is present, it binds to the lac repressor protein and triggers repressor to release DNA. This induces transcription. So typically for the lactose operon, the repressor is already attached to the operator. So RNA polymerase cannot bind and will not transcribe the genes. But when lactose becomes present, it will bind to the repressor. It'll have a conformational change in the repressor protein, thereby removing the repressor, allowing for RNA polymerase to bind, transcribe the gene, and create mRNA. The mRNA is then going to transcribe enzymes that will then digest the lactose. So what happens when lactose is present? Unlike the tryptophan, where the enzymes are no longer being produced, when tryptophan is present, it needs to make lactose, when lactose is present, there needs to be lactose digestion enzymes produced. So it turns on transcription and allows for enzymes to be made to break down the lactose that is present. Lactose is also an allosteric regulator of a repressor protein. So Jacob and Manad studied the lac operon. They were first to describe the operon system, and they coined the phrase operon. So to summarize, we have repressible operons and inducible operons. And a repressor operon usually functions in an anabolic pathway, meaning it stores energy. It's synthesizing end products. So when product is present in excess, the cell allocates resources to other uses. So instead of wasting energy to produce more, it puts a stop to it and holds off until it needs to produce more because there isn't any of that product present. Inducible operon usually functions in a catabolic pathway, meaning releasing energy or breaking down more complex molecules. So our example would be lactose. When lactose is present, lactose digesting enzymes will become present to break down the lactose to a simpler molecule. Produces enzyme only when nutrient is present. And the cell avoids making proteins that have nothing to do, so the cell allocates its resource to other uses. Don't be repressed. How can I induce you to ask questions? Please post your questions on Schoology so I will be prepared to answer them at the next class. Have a good night.